students, my name is Niyati Said and thanks for watching Edupedia Word Videos. My topic for the presentation is the fifth section of the chapter Excretion and Osmoregulation. That is, excretory products and their elimination. In this presentation, we'll be studying about the three detailed steps of urine formation. Okay, so in my previous presentation I have already taught you that the formation of urine takes place in three steps, right? Which are those? Ultrafiltration, selective reabsorption and tubular secretion. Okay, and I have already taught you glomerular filtration also. So the glomerular capillary blood pressure, it causes filtration of blood through the following three layers, which are those: endothelium, epithelium and basement membrane. Now the endothelium of glomerular blood vessels, epithelium of Bowman's capsule and the basement membrane between these two layers. Which two layers? Endothelium and epithelium. Okay, so these three layers they help in the glomerular filtration. Okay, or you can say that glomerular capillary blood pressure, it causes filtration of blood through following three layers: endothelium, epithelium and basement membrane. Okay, see, increased glomerular hydrostatic pressure 55 mmHg, increased blood colloid pressure which is about 30 mmHg, and capsular hydrostatic pressure which is around 15 mmHg. Okay, epithelial cells. Epithelial cells means podocytes. Podocytes are present in Bowman's capsule. Okay, so the epithelial cells, they are arranged in an intricate manner that leaves some minute spaces which we call it as filtration slits. Okay, see, these are filtration slits, or you can say that when the podocytes they are arranged in intricate manner, they leave some minute pores. These are the minute pores which we call it as filtration slit, and they are also called as slit pores. Okay, and this yellow one is podocyte and this is podocyte body. Okay, almost all constituents of blood plasma except the protein pass on to the lumen of Bowman's capsule. Okay, and about 1100 to 1200 ml of blood is filtered by kidneys per minute. Okay, per minute they filter about 110 to 1200 ml and blood is filtered by kidneys per minute, and it constitutes 1/5 of the blood pumped out by each ventricle of the heart in a minute. Okay, so you can say that glomerular filtration is also known as ultrafiltration. It is a physical process of filtration of blood under high pressure that takes place in Malpighian body. Malpighian body, I have already taught you. Okay, what it is. It is also known as Malpighian corpuscles. It is the anterior part of the nephron situated in the cortex. What is nephron? Nephron is a basic structural and functional unit of kidney, and it is about 40 to 60 nm in length. And so now coming back to ultrafiltration. Ultrafiltration is also known as glomerular filtration, students, so don't get confused with it. In this what happens is Bowman's capsule. See, this is Bowman's capsule. Okay, so it acts as a, you can say that it acts as a ultra filter. The main structure involved are visceral epithelium of Bowman's capsule and glomerulus. The wall of glomerular capillaries is made up of single layer of cells which we call it as endothelium, which I have already taught you. Okay, and The walls of the capillaries is fenestrated. Fenestrated means porous. See, these walls are porous. Okay, that is, it has number of pores which allow most of the constituent of blood plasma to pass except plasma proteins and blood cells. Okay. The epithelial cell of visceral epithelium are modified and they are called as podocytes. Podocytes I have just taught you, and they give out a foot-like processes called pedicels. In the previous diagram, let's see. This is podocyte. Okay, so the podocyte it has number of pores which and it gives out foot-like processes which we call it as pedicels. These are the secondary processes. Okay, the pedicels, they are interdigitate so as to leave fine spaces called filtration slits. Okay, the slits form the filtration path as they are in intimate contact with basement membrane of endothelium of glomerular capillaries. Okay, I repeat, these filtration slits, they form a filtration path as they are in intimate contact with the basement membrane. See, this is the basement membrane, this red one. So it is in the intimate contact with the basement membrane, so of the endothelium of the glomerular capillaries. So these slits, they form a filtration path. Okay, filtration path as they are in intimate contact with basement membrane of endothelium of glomerular capillaries. This one, the red one. Okay, now coming back to our topic. Okay, so the afferent arteriole, this is our afferent arteriole. Okay, the afferent arteriole, you can say it is a branch of renal artery that enters the Bowman capsule. See, it is entering the Bowman's capsule and it forms a knot of capillaries which we call it as glomerulus. So this is not like capillaries which we call it as glomerulus, or you can say that it is a tuft of capillaries, so we call it as glomerulus. The blood, it leaves the glomerulus through efferent arteriole. See, the blood is leaving through efferent arteriole. The diameter of afferent arteriole is wider than that of the efferent arteriole. Okay, please note that. And hence the blood in the glomerulus is under pressure because the afferent arteriole of the glomerulus has a greater diameter in comparison to efferent arteriole. Okay, now a question may come that afferent arteriole of the glomerulus has a greater diameter than efferent arteriole. So you can say that afferent arteriole, it supplies blood to the glomerulus. Okay, see, it is entering and thus the blood is also entering through afferent arteriole. So the afferent arteriole, it supplies blood to the glomerulus, and efferent arteriole, it carries blood away from the glomerulus. Okay, it is taking away from the glomerulus. And second point, you can say that as the afferent arteriole is wider than efferent arteriole, the amount of blood coming into glomerulus is more than the amount of blood drained out of it. Okay, so this is the second reason why the afferent arteriole has a greater diameter in comparison to the efferent arteriole. Now the third point, you can say that the difference in the diameter creates high blood pressure. Okay, in the glomerulus, due to which blood is filtered. And that's the main function of kidneys, to filter the blood. Okay, so the difference in the diameter of afferent arteriole and efferent arteriole, it creates high blood pressure inside glomerulus, due to which blood is ultra filtered. And due to this reason, afferent arteriole has a greater diameter than efferent arteriole. Okay, and about 1100 to 1200 ml of blood is filtered by kidneys per minute. It constitutes 1/5 of the blood pumped out by each ventricle of the heart in a minute. Now, the amount of the filtrate formed, the ultrafiltrate amount formed per minute is called as glomerular filtration rate, which we call it as glomerular filtrate rate, that is GFR. Okay, or you can say that GFR is also known as primary urine or deproteinized plasma. Okay, why this GFR is also called as deproteinized plasma? Because of the pressure, the blood that passes through the capillaries of the glomerulus is ultrafiltered. During the ultrafiltration process, the blood cells cannot pass through the pores, and hence only the blood plasma is filtered. Practically all the components of plasma except large molecules of plasma protein pass through the pores and they appear in the cavity of Bowman's capsule. And that's the reason this glomerular filtration rate is also known as deproteinized plasma. It has both useful material such as glucose, amino acids, salt, as well as waste material such as urea from the blood. So the filtrate, that is GFR, is isotonic, or you can say that it is similar in composition to blood plasma. Okay, and please note one thing. This ultrafiltration, or you can say the glomerular filtration, is passive in nature. That means it doesn't require energy. Okay, so this ultrafiltration is passive in nature and non-selective. Okay, and the rate of the ultrafiltration is 180 liters per day. Okay, and you will be amazed to know that amount of plasma in body is about 3 liters. Thus, every drop of plasma is examined by kidney about 560 times per day. Okay, and please note one thing, that the rate of filtration, which we call it as GFR, that is glomerular filtration rate, is about 125 milliliter per minute. That is about 180 liters of filtrate is formed by kidneys in 24 hours. Okay. So, a glomerular filtration, it must be precisely controlled. If it is too high, fluid flows through the renal tubules too rapidly for them to reabsorb the required amount of water and solutes. And if it is too low, fluid flows too slowly through the tubules, and waste that should be eliminated are reabsorbed into the blood stream. Okay. Now, what is renal autoregulation? Renal autoregulation is the ability of the nephron to adjust their own blood flow. That's why it is called as autoregulation. It flows them to maintain a relatively stable GFR in spite of changes in arterial blood pressure. Okay. Now moving on to forward with the second stage of urine formation, that is reabsorption. Okay. Now what happens in reabsorption, students? It is also known as selective reabsorption. Okay. So in this, what happens is it is a process of reclaiming water and other substances from tubular fluid. Okay, and then it returns them to the blood. We have just studied ultrafiltration or glomerular filtration that has formed a glomerular filtrate. So the glomerular filtrate formed in the urinary space after ultrafiltration, it contains nitrogenous waste as well as useful substances such as salts of sodium, potassium, calcium, chloride, vitamins, water, glucose, etc. So the useful substance need to be reabsorbed in the body, right? So the glomerular filtrate passes through the renal tubule. Okay, it passes through the renal tubule, where selective reabsorption of useful substances takes place from renal tubule to the blood in the peritubular capillaries. Okay. And the renal tubule, it comprises of PCT, that is proximal convoluted tubule, Henle loops, distal convoluted tubule, and terminal duct. I have already taught you this. Okay, students, you can refer to my previous presentation. So the renal tubule, it comprises of proximal convoluted tubule, Henle's loop, distal convoluted tubule, and terminal duct. Okay. And the inner lining of renal tubule is made up of columnar, cuboidal, ciliated epithelium, which has microvilli. Microvilli, they help to increase the surface area for reabsorption. And the cells, they have many mitochondria to provide energy for active absorption. So the process of reabsorption takes place by active transport, passive transport, and osmosis. Okay. Now let's see how sodium reabsorption takes place. So the sodium reabsorption is the key to everything else. It creates the environment for water and other substances to be reabsorbed. The useful substances I am talking about. Okay. Now the glucose. Glucose is transported along with sodium ions by carriers called sodium glucose transport proteins. So this sodium glucose transport protein, they help sodium ions to be transported. Okay. Normally all glucose in the tubular fluid is reabsorbed and there is none in the urine. Okay. Now coming to the water reabsorption, as I have told you that water also gets reabsorbed. Okay, so the water reabsorption is a significant function of the kidney. The amount of water reabsorption is continually regulated by hormones according to body state of hydration. The more hydrated the body is, the less water is reabsorbed, and opposite is also true. Okay. Now coming to reabsorption, I have told you that reabsorption is also known as selective reabsorption, and it takes place in renal tubule, and renal tubule comprise of PCT. This is PCT, okay, proximal tubule, and loop of Henle, and distal tubule, and terminal duct. So the renal tubule starts from here to here, okay, till collecting duct. Okay, so you can say that the tubular fluid, okay, the tubular fluid, it passes down the descending, this is the descending limb of the loop of Henle. So the tubular fluid, it passes down the descending limb of Henle's loop. The descending limb is permeable to water. See, this is permeable to water. Okay. But almost impermeable to electrolytes. It doesn't allow any electrolyte to pass through it. And water is progressively reabsorbed when filtrate passes down the descending limb, and hence the filtrate becomes progressively hypertonic. In the hairpin bend, this is hairpin bend, okay, we call this as hairpin, okay, because it resembles like hairpin, and this is hairpin bend. So the hairpin bend, it is maximum hypertonic, you can say. Okay, and now comes ascending this is ascending or the thick segment of the ascending limb. Okay, and now ascending limb, you'll be like amazed to know that it is permeable to electrolytes, unlike descending limb of the loop of Henle, but it is impermeable to water. It is just opposite. As the filtrate passes through the ascending limb, the electrolytes, they are reabsorbed actively or passively into medullary fluid. Okay, this is the medullary part, and all the electrolytes, they are reabsorbed actively or passively into the medullary fluid, and the filtrate becomes hypotonic. See, the filtrate in this descending limb was hypertonic, but when it comes here, it becomes hypotonic. Okay. Now the filtrate comes to distal convoluted tubule. This is distal tubule. Okay, so the distal tubule, now the filtrate comes to the distal convoluted tubule, where water is reabsorbed by osmosis, and Na+, K+, and Cl- and HCO3- are reabsorbed. Okay, now comes collecting duct. This is collecting duct. Okay, this entire tubule is collecting duct. So in the collecting tubule, some more water is reabsorbed due to presence of Na+ and Cl- in the surrounding tissue fluid. This increases urea concentration in the collecting tubule, and as a result, some urea, it diffuses out passively from collecting tubule. While passing through collecting tubule, the urine becomes hypertonic. That means when the filtrate comes here, it becomes hypertonic. Okay, the amount of the glomerular filtrate formed per day is 180 liters, and the urine released per day is about 1.5 liters. This suggested that nearly 99% of filtrate is reabsorbed. Okay, this is what the second step takes place, or this is how the selective reabsorption, that's the second step of the urine formation, takes place. Okay, and please note that substances like glucose, amino acid, Na+, in filtrate, they are reabsorbed actively, and nitrogenous waste, they are absorbed by passive transport. Okay. And one more thing, that the passive reabsorption of water occurs in the initial segments of the nephron, I have told you. And PCT, this is PCT, okay, proximal tubule, that is proximal convoluted tubule. So this is PCT. It reabsorbs most of the nutrients and 70 to 80% of the electrolytes and water. Now, why does it has power of reabsorption, students? That's because of the PCT, because PCT has simple cuboidal brush border epithelium that increases the surface area for reabsorption. Okay. This I have already taught you, the loop of Henle. This is the descending part and this is the ascending part. So the descending part, minimum reabsorption takes place and it maintains high osmolarity of medullary interstitial fluid. Whereas the descending limb, it is permeable to water but almost impermeable to electrolyte, and this concentrates the filtrate. And whereas this ascending loop of Henle, it is impermeable to water but it is permeable to electrolytes. It is just opposite to descending lip of loop of Henle. So the filtrate gets diluted. Okay, I have already taught you this. Now the filtrate comes to the distal convoluted tubule. This is distal convoluted tubule, where conditional reabsorption of Na+ and water takes place, and then comes collecting tubule. This is collecting tubule. It extends from cortex. As you can see, this is cortex, outer medulla and inner medulla. So it extends, this collecting tubule, it extends from cortex to inner medulla, and it reabsorbs large amount of water to concentrate urine. and it allows passage of small amounts of urea into medullary interstitium to keep up the osmolarity. And now comes the third process, which we call it as tubular secretion. The third process by which kidney clean or filter blood is called as tubular secretion. This is tubular secretion, and it involves substances being added to the tubular fluid. What happens in the third process of the urine formation, that it removes excessive quantities of certain dissolved substances from the body, and it also maintains the blood at a normal healthy pH level. Okay, what is the range of that pH? It is around 7.35 to 7.45. okay. The substances that are secreted into tubular fluid for removal of the body include potassium ion, hydrogen ion, ammonium ion, and creatinine urea, and some hormones and some drugs also. So the tubular secretion, which is the third process of the urine formation, it occurs from the epithelial cell that line the renal tubules and collecting duct. Now comes tubular secretion, which we'll be studying in detail, that it is the third step in urine formation. Although the blood is ultra filtered in Bowman's capsule, but the some amount of blood escapes ultrafiltration, which is the first step of the urine formation. Okay, so this blood, the remaining that blood which escaped ultrafiltration, it circulates in capillaries around the renal tubule. Okay, and it contains nitrogenous waste such as urea, drugs, and some ions such as potassium ion and hydrogen ion. Okay, so from these capillaries, I am talking about the red one, students. The red capillary, the nitrogenous waste like ammonia, urea, creatinine, as well as some ions such as K+, H+, they are absorbed actively by distal convoluted tubule. Okay, or you can say that they are absorbed actively and they discharge in the cavity of the renal tubule. Thus, this process is just opposite to selective reabsorption, which is the first step in urine formation. Okay, and note one thing, that the secretion of hippuric acid, creatinine, ammonia, and certain drugs, they take place in PCT. But the PCT also helps to maintain the pH and the ionic composition of body fluid by selective secretion of hydrogen ion, ammonia, potassium ion into filtrate, and by absorption of HCO3 minus ions. And you can say that the collecting duct, it maintains pH and ionic balance of blood by the secretion of H+ and K+ ion. Now, what is the role of collecting duct in the third stage of urine formation, that is tubular secretion? The role of collecting duct is that collecting duct is a long duct that extends from cortex of the kidney to the inner part of the medulla. So large amount of water could be reabsorbed from this region to produce a concentrated urine, and this segment, it allows passage of a small amount of urea into medullary interstitial fluid to keep osmotic concentration. So you can say that collecting duct, it plays a vital role in maintenance of pH and ionic balance of blood by selective secretion of H+ and K+ ions. Okay, so this is the three steps that are involved in urine formation. This is afferent arteriole, this is efferent arterioles, and this is Bowman's capsule. And what is this? This is glomerular capillaries. So this is how it takes place. First is filtration, that is ultrafiltration, which we have already studied. Then comes the selective reabsorption. Okay, And then comes secretion, which is the third step in urine formation. So this is how 3 steps are involved in urine formation. Okay. Now we'll study the concentration of the filtrate. Now we know that the glomerular filtrate in the Bowman's capsule is deproteinized plasma and hence is isotonic to blood plasma. Okay, in the proximal convoluted tubule, maximum about 65% of the reabsorption takes place. As a result, after passing through PCT, the filtrate is hypertonic. Even while passing down the descending limb of the loop of Henle, the filtrate becomes hypertonic as water is reabsorbed. And in the ascending limb of Henle's loop, however, the filtrate loses NaCl and becomes hypotonic to blood plasma. And in distal convoluted tubule, the filtrate again becomes isotonic or hypotonic blood plasma because first aldosterone causes reabsorption of Na+, and second ADH causes reabsorption of water. So this hypo or isotonic filtrate is now discharged into a common collecting tubule, which is now called as urine. However, before it is discharged out, it is made hypertonic by reabsorption of water, and thus hypertonic urine is discharged. Okay, and the flow of filtrate in the 2 limbs of Henle's loop and the flow of blood through the 2 limbs of vasa recta are in opposite direction, which we call it as counter current system. Counter current pattern or system is composed of loop of Henle, vasa recta, which is present in the medulla part of the kidney. Okay. So the counter current and the proximity between Henle's loop and vasa recta maintain an increasing osmolarity towards the inner medullary interstitium, that is from 300 more small liter inverse in the cortex to about 1200 in the inner medulla, and this gradient is mainly caused by NaCl and urea. Okay. As you can see that this is the descending loop of Henle and this is the ascending loop of Henle. So the descending and the ascending limbs are anti-parallel to each other. So the filtrate in these limbs moves in opposite direction. Of course, it moves in the opposite direction, and hence the name is counter current. The Henle's loop is surrounded by medullary interstitial fluids. The fluid that surrounds the loop of Henle is called as medullary interstitial fluid. Okay, and the medulla also contains vasa recta as part of peritubular capillaries from efferent renal arteriole. The vasa recta also consists of 2 limbs, which are those descending and ascending. The blood in these 2 limbs of vasa recta also flows in opposite direction. The descending limb of vasa recta, this is the vasa recta. The descending limb of the vasa recta is very closer to the ascending limb of the loop of Henle and vice versa. So the descending limb of Henle's loop, see, this is the descending limb of loop of Henle's. It is permeable to water. I have already taught you that it is permeable to water, but impermeable to electrolytes. While ascending limb of loop of Henle, it is just permeable to electrolytes but impermeable to water. And now coming on to descending loop of vasa recta, this is vasa recta, okay, and this is descending loop. So the descending limb of vasa recta have walls with water channels that has aquaporins, and the ascending vasa rectas, this is ascending vasa recta, have endothelium with wide gaps between cell that allow rapid diffusion of solutes. Okay, and this is how the counter current system is responsible for concentration, that is hypertonicity of urine when it passes through the common collecting tubule. Okay, this I have already taught you. Now that is same counter current mechanism, how it is facilitated. Okay, and the human kidneys produce urine 4 times concentrated the initial filtrate formed. Okay, the relative concentration of the urine depends on the body state of hydration. For example, if you drink a large volume of water, you will produce a large volume of urine, and that is very less concentrated. On the other hand, if you are dehydrated, that means the water quantity is very less in your body, so your urine is much more concentrated and the volume is much lower. The collecting duct can adjust water reabsorption, that depends on the body's need for water conservation or removal. Okay, so these were the 3 steps that were involved in urine formation. Okay, so I hope it's clear to all of you. Thank you and keep watching Edupedia word videos.